Hello, I'm Jianhong. Today I will introduce TrackViewer, a tool to do Lollipop Dandelion plots for methylation and mutation data. Before we start, I suggest you install the workshop package from GitHub. The package contains the vignettes and the slides for this workshop. First, set your working directory to a new temporary path. When the workshop finished, you can remove all the files generated in this workshop by removing the temporary folder. We use install function from Bulk Manager to install the workshop package. It will take a lot of time to generate the vignettes, so please do it as early as possible. Here, I list some useful links in red. They are the links for source code, vignettes, and slides for this workshop, the link for TrackViewer, and the publication for the package. There are two kinds of data I want to involve in today's workshop: methylation and mutation data. Epigenetics is the study of mechanisms that produce phenotype changes without alternation in the DNA sequence. It includes DNA methylation, histone modification, and non-coding RNA. There are multiple types of DNA methylation. For example, 5-MC is a methylation of the 5 position of a cytosine. The DNA methylation can control transcriptional expression. Usually, methylated DNA will recruit some protein complex to repress the binding of RNA polymerase, or TFs, which in turn repress the gene expression. For example, if the promoter region of the tumor suppressed gene is methylated, as shown hypermethylation here, the expression of tumor suppressed gene will be downregulated, which in turn lead cancer happen. Like UCSC Genome Browser, Washington University provides an epigenome browser. It is very similar to UCSC Genome Browser. It can also show the interaction in different chromatin regions. Another type of data I want to involve is site mutations. Mutation is basically an induced misspelling of the DNA sequence. There are several types of them: point mutation, chromosome mutation, and copy number variation. For chromosome mutation, there are inversion, deletion, duplication, substitution, and translocation. For point mutation, there are SNPs and insertion, deletion. We also call it indels. There are lots of well-known databases contain the mutation informations. For human, Cosmic Database is an online genome browser to view the point mutations. dbSNP also contains the mutation information for human. Beyond the online tools, there are many tools to create mutation diagram. Methylation, mutation, and SNP data require special type of plot called a Lollipop or needle plots. The most famous tool is ChIP-seq Mutation Mapper or PFAM lollipops. They are the most widely used tools for DNA methylation, DNA mutation, and protein mutation plot. There are so many choices. Why TrackViewer for Lollipop plot? Just because it is beautiful. It is a minimized design, simple, clean for publication, and it is powerful for high-dense data. The shortage for this tool is that you must be familiar with R, and there is no embedded database. That is to say, you need to prepare all the data for plots by yourself. Here is a list of current available tools to call SNPs indels. We can see that for most of them, the output is VCF file. Here is an example of VCI file. The VCI file has a chromosome, set mutation position, ID, reference base, mutation base, score of the mutation, filter status, and additional information. The explanation of the additional information is listed in the head section. VCI file, byte files, and their variations can be the input files for plots. They are the most popular file formats to hold the methylation and the mutation data. To extract the annotations, you can use TXDB, EnsemBleDB data, Annotation Hub data, and other online resources which can be converted to GRENS object. By combining the annotations and methylation or mutation data, TrackViewer can plot Lollipop plots for low-dense events and the Dandelion Plot for high-dense events. TrackViewer can also combine the coverage plots with the Lollipop plots and generate the integrated plots. For Lollipop plot, it is highly customized. You can change the labels, symbols, colors, opacity, and size. You can plot the scores in stacked symbols or pie chart, even the shape defined by yourself. In addition, we implemented a web application of TrackViewer, leveraging Qian package. The web application of TrackViewer is available at this address, listed in the bottom of this slide. Today, I will not show the app. You can get more information from the videos in our publication. Here are some examples in publications that people use in TrackViewer to show their data. In left figure, the authors are comparing the missense variants in the regions of ACE2 gene from male to female. In the right figure, the authors are using Lollipop plot in pi stack layout to represent SNPs in different patients. In left figure, you can see the no color indicates the missense types. There are counts in the nodes. It contains lots of details, but not busy. In the right figure, the authors using colors to show the base for each patient. Indeed, the TrackViewer asks all inputs for Lollipop plots in GRENS object. The GRENS object contains sequence names, start, and the end position, the strand, the information for chromosome length, and additional information. We use additional information to control the Lollipop plots. I put all the vignettes for this workshop online now. You can go to this website to see the vignettes. You can start to use Docker to pull down the last image of this workshop. First, try to make sure your Docker is running. And then you can see Docker pull Jianhong TrackViewer Elk Asia 2020 workshop latest. It will pull down to the Docker image. We can run the Docker. First, we change our directory to the home folder, just in case there is some issue with your Docker. So then we can create a temperature path, make directory temp for, I call this name as TrackViewer. And then we can use docker run to run the docker. So docker run. We give a password to the RStudio. We set the password as 123456, and forward the port E787. This port is used by the RStudio, and then we bound the folder we just create called temp for TrackViewer to the container volume data folder, and then we supply the Docker image name. It will run. Once we got it run, Web Explorer, use localhost E787 to access the Docker container. We use RStudio as username and the password 123456. This is a password we just set. As we can see here, this is the online weakness. Here is a hyperlink for the source code. Here is a hyperlink for the slides. If you click here, go to article, then you can see the weakness for the TrackViewer of this workshop. Here is a table for available tools for Lollipop plots. Because in the Docker we already have everything installed, so we can directly start the code parts. We do not need to install the TrackViewer. First, let's check the package version. Of TrackViewer. And then we can start our plot. Today our job is generate one Lollipop plot and one Dandelion plot. So load TrackViewer package, and we can check the parameters for Lolliplots. I ask help. We got a lot of parameters: SNP, GR, futures, and ranges, and so on. Let's see what should we prepare. SNP-GR is the G-range represents the mutation and methylation data. Here we can start some data, say CHR1 as seq name, and I range the star site we set as C10, and another one set as we set 20 and 150, and we set the y's equal to 1 and give the names as SNP1, SNP2. Let's see what we got. It's a SNP1. In the G-range object, we have 2 ranges, 0 metadata. The seq name is chromosome 1. The range start 2050. The name is SNP1, SNP2, okay? And then we assign filters. We generate some manual 1 G ranges as filters, keeps the seq name same, and size the range as I ranges. It says start from, for example, from 1 is from 10, another from 80. The end side is 50, 20, and 160. And then give some name. Say the first one is a 5 UTR. The second one is CDS1. And then we can do Lollipop plot. Lollipop. We got our first plot. You can see the mutation SNP data is shown here. We want to get wider range. Wider view, we set the range parameter as a G range, say CHR1. We give start position 1 and end position, for example, 200. Then we get wider view. Now we want to change the filter color and height. We set the filters color by field, the metadata field. We see it light blue and dark blue. And then we check the filters. We can see there are 1 metadata called field. And then we plot the data. You can see the color already changed. And we want to change the height. We set as, we use the full height of the canvas is 1. We set it 0.025. The second one is 0.05. And then we check about the height. We can see the height changed. And the next step, we want to change the SNP score. We set some random data. We see we get more SNPs. We set more SNPs to show the lollipops. We set SNP. The position is the view position is 200, so we sample number from 200. We set 20 SNPs. We assign the mutation data. the start equal to SNP, and y is equal to 1, and the names we just paste prefix with the SNP. Then we can check what we got, and we can plot the data. We can see a little busy here, and we assign the score by random number. We set as 10 because we assign the size total is 20. We also say keep same here. We must size replace equal to true, otherwise we will get an error. Then we see, okay, we have a score column here, and then we do the plot. Okay, we get some plot like this. Okay, reassign our score number. We set the score number example from the max value is not 10 anymore. We set its max value as 100 and the size keeps same 20. And then we do the Lollipop plot again to see what we got. The figure is something like this. It's not a stacked circle anymore. And then we want to change the color of the plots. We say we change the color by direct assign a color to the mutation data. We say the color examples max number is 8, and then the size is 20. Replacement because the max number is smaller than size, we must set as true. And then we check the color column is there, and then we do Lollipop. You can see the color already changed for each node. We can also change the color alpha value by set alpha. We set alpha from 100 to 55, and the size is 20. Convert it. Oh. You can see alpha value is applied. We can also add some label to each end. We see a label to each end. We set the label as the mutation name. can be set as something must be a character. So we set as character. We will see some number in the node. We can also change the label color. See. If the node color is black, then we change it to white. Otherwise, black. It looks like now changed. It happens there is no color equal to 1. Then we change to, for example, we change it be equal to 8. We set it as white. See, you can see this one change to white. We can also add some legend. The names of the legend will be the labels. You can see the legend is already plotted here. If we want to change the plot into a pie charts, we need at least 2 scores. We set the score 2. We also sample some number. And the size equal to 20, the length of the mutation. Check is pi. It already changed, but we see the color is same here. We need to change the color of them to a list. We see we already have some color set. We just change it to a different color. We give a 0. We see what we got. We have list of color of them. The first always 0. We can see there is always white color here. If we want to change the plot something like a caterpillar layout, we just add SNP side ID. Wait. The SNP side must be top and bottom. Top or bottom. Size is 20. Replacement must be true here. Plot it again. We can see the style already changed to caterpillar layout. The next step is we want to plot if we have more of them. For example, we have 50 of the mutations. We see example into we have maximal 20. We get 40 of them. If we still use we plot by Lollipop, it will somehow a little busy here, right? So if then we can change to use a Dandelion Plot. The parameter almost same. We must set the type equal to circle. For this example, if you want to use a fun, you at least give a 2 score for the data. We can get the Dandelion plots. As a summary, TrackViewer can generate Lollipop plot to show the methylation and the SNP mutation status, visualize regular race coverage tracks. It can be easily integrated into standard analysis pipeline for high-throughput sequence datasets such as ChIP-seq, RNA-seq, methylation-seq, or DNA-seq. It can produce highly customized figure for publication. I would like to thank my previous PI Julia Zhu, my current PI Ken Pos, and my previous colleagues Junyu, Haibo, and all the lab members in Pos Lab. Thanks for Amy Dixon for running Regeneration Next. Thanks for regional omics shared resources. Thanks for Committee of Biochem Asia 2020 give me the opportunity to present the TrackViewer package. Thank you for your attention.